neutrophil, but if they put it in context, you can see the really dark red coloration. And these guys have that weird specialized job of killing parasitic worms. Basophils tend to be smaller, tend to be really cloudy. This one, the basophil and the lymphocytes, sometimes can be confusing, but look for this little clear patch on lymphocytes, because you're always going to see that. They don't have granules, and their nucleus doesn't take up the whole cell, so you get to see like a little crescent. Basophils are generally just completely dark, and they're not really to scale. Look at how big this is compared to these blood cells compared to that guy. And this is a monocyte, big kidney bean shaped nucleus, also a phagosome. I'm getting a lot of walking here. All right. So know the blood cell types. Again, there's a lot of disorders. Save that for the end, right? That's just icing on the cake. If you've got time at the end, you're like, what should I do now? Go study those disorders, because there's not going to be a bunch of them. Blood typing. This is something that a lot of students get confused about, I think. So I'm wagering that not everybody in here knows exactly what blood typing is all about. So I'm going to give you a quick little tutorial on it. Unless you don't want me to. No, do it? Do it, okay. All right, so you basically, here's a red blood cell, and it doesn't really have a little letter A on it, but it is got a chemical found on the plasma membrane on the outside, which people that discovered it have called it they call these things agglutinogens. And a agglutinogen, if you break down that word, it generates agglutination, so it's going to cause clumping. And this is something that they discovered when they started transfusing blood, right? When you got blood transfusions, some people were fine with a pint of blood being given to them. Other people would suffer, death from it. So they figured out that they better figure out what's wrong with these different types of blood. So blood can either have the A agglutinogen, it could have the B agglutinogen, or it could have nothing of these three, right? And so they called these the A group, the B group, and the O group. Or zero, really. It means nothing. So A is something, B is something, type O is lack of something, right? It's not having something. Now this is the ABO groups, and over here we've got the RH, which is either yes or no, it's either have it or don't. So blood type RH negative isn't a thing, it's just you don't have anything. So you can either have the RH factor, or you cannot have the RH factor. So it's presence or absence. And these are different traits, so you can combine them. You can be, if you've got the A, the B, and the RH, well, that means you're AB positive for that RH group. Now, the way our immune systems work and the way those blood type tests work is that if I have something, let's say that I was AB positive. I'm not. If I was AB positive, and so-and-so, who knows their blood type? What do you got?
O negative.
O negative, me too, actually. But if I were, good job, we have valuable blood. So, all right, let's go with O positive. So is there anything on her blood, on O positive, that I haven't seen before? What about the O?
It's okay, it doesn't have a marker.
Right, O is nothing, right? Nothing isn't something. Nothing isn't something. That's a pretty good way to say that. So the O is a nothing, just like the negative's a nothing. And some people with the O or with the negative, doesn't affect who they can donate to at all. So if I'm AB positive, I've seen the A, I've seen the B, and I've seen the positive. These two are nothing. What if I were B negative? Whose blood could be given to me? You can always give yourself, right? B negative. You can always donate to yourself. And O negative, but O is different. O is nothing, no chemical marker, right? So if it's either an O or negative, those don't affect who you can donate to, right? So anybody can always take the negative of whatever it is they have. And anybody can always take an O of whatever it is that particular thing. Now for us two, and for several other people probably in here, who are O negative, my blood doesn't have an A, doesn't have a B, doesn't have the RH factor, right? So anything except for O negative is new to me. O positive, hey, O sounds safe. Positive is that protein, and I'm going to react adversely to it, right? So there's tables out there you can look up who can donate to who and who can receive from what. But suffice it to say that this one is what they call the universal donor, because it's a blank slate, and this one over here, AB positive, is the universal recipient, because there's nothing out there that it hasn't seen. Now when it comes for those blood type tests, there's a couple of ways to do them. I'll either use a photograph, or probably a photograph of these things, or a representation of one, but whatever. We had on the fake blood ones, which I don't know if you've seen them, but the fake blood ones had like an A, a B, and an RH. They might have called it D or something. So I'm going to put blood in here, blood, and then I'm going to add anti-A to this one. So if the blood has A, you're going to get coagulation. Add anti-B to this one. If the blood has B, you're going to get coagulation. Add anti-O to this one. So if the blood has, anti-RH factor to that one. If the blood has the RH group, you're going to get coagulation. So if I end up with smooth blood, clumpy blood, what do I got? Deep. I tried. But this is anything that stands out, right? Anytime you get a reaction, that indicates the presence of that particular marker. B positive joke?
Yeah, I got it.
Now, if you guys remember those, did you guys do your own blood types?
Yes.
Some classes did, some classes didn't. We had 4 things. This was the control right down here. So this should have always been just clear blood, right? Otherwise, it's the same. I think it was A, B, RH, like this. Something like that. Doesn't matter what the orientation is. But these little speckles would form, right? Or you'd get smooth blood. What do we got? AB negative. So it's really straightforward as long as you just know what you're looking for. If it looks like clean blood, there was no reaction, means it doesn't have that particular marker. If the blood clots up and it's in the B cup, well, it's got the B agglutinogen. Does that make sense? Yeah. So as long as you can, there's tables out there you can just look up, like blood donation table or something. Type that in. You'll probably get a table that'll work. Okay, so blood typing, no worries. Check. Let's go to the heart. Now, the list of slides here, there's, in my slide box that I'm looking at right now, which is the same one you got, the same one they have in the Science Learning Center for the most part. We've got 7 endocrine system. We've got 1 basic blood, and then 2 or 3 blood disorders, right? So you probably get one of those on there. Then we go into the heart and blood vessels. So there's 1 slide of cardiac muscle, which you're looking for what? Intercalated discs. So if you see blood, and you see a heart muscle with striations and maybe some branching, which you might be able to pick up, and then another cell over here, and there's a dark line there. We're not going to be looking at skeletal muscle. You might see some smooth muscle, but you're going to be. That indicates that that's cardiac muscle. Now, as for the heart itself, gotta make my way. Don't worry, you don't have to film me. I'm going to go back to the front. No? Somebody tried to make a baby heart out of 2 fronts. This will work out. We can Frankenstein this one together. All right, this is fine. So, here's a heart. This is a big heart model. You're going to see. I'll go behind the velvet roof again. So here's a heart model. You're going to see this out there once or twice or 3 times, right? You're going to get that cow heart out there, right? But what you want to look for, you get your basic orientation down. You want to know which sides, and it's not really obvious here which side's thicker, but on the real heart it is. And on this side, if you're looking at this thing from the anatomical position like this would be placed in my body like this. So my left is your right, right? So this side is the left side of the heart. It's a little tricky. but just take your time, you'll be okay. So I'm gonna open this up. So if I know this is the left side and this is the right side, well, I can get a bunch of landmarks down now, right? So if this is the left side, this is the right side, then this is the right atrium, right ventricle, left atrium, left ventricle, pretty easy. The valves are important because they're different, these two valves right here. If you wanted to say, if you forget which one's the bicuspid and which one's the tricuspid, or which one's the mitral and which one's the tricuspid, you can just say right atrioventricular valve or left atrioventricular valve. You can say that, that's fine, right? But if you want bicuspid and tricuspid, the way I teach my class this, and I apologize for being repetitive, is that your left side of your heart is the strong side because it's got to pump out to your body, right? Left side also has the bicuspid valve. So the strong side has the bicuspid valve. If you ask your little nephew to show you his muscles, which muscles does he show you?
Biceps.
The biceps, right? So he's going to go, these ones, right? And so the biceps and bicuspid are strong. Left side. Unless he shows you like a lat spread. Lat spread, of course. Straps, I don't know. So there's some valves you have to know, and you have to know the path of blood through the heart. Now here's one thing that a lot of students get confused about, and I want to make sure that you guys don't mess this up next week if it's on the test. These little tendinous cords right here. Chordae tendineae, right? As soon as you get to the bottom and you hit this meaty stuff, those are papillary muscles. Okay? So the chordae tendineae are just the fibers and the cords. The valve is not chordae tendineae. The valve separates the atria from the ventricles. The valves attach to the chordae tendineae. And it looks like those chordae tendineae would be like drawstrings to open up the valve. They are not. Just gravity is going to push the blood from the atria to the ventricles. When these ventricles contract, they're trying to push blood out through the pulmonary artery and through the aorta. That's where they want the blood to go. But there's two chambers right up here that the blood could go back into were it not for these valves. So the valves prevent backflow into the atria, and the chordae tendineae keep them from prolapsing, from flipping the other way. So they're just kind of restraining cords. They're not pulling to open. They're just tightening up to prevent the blood from going up in there. All right. The blood goes down to these blood vessels. Now another thing that's confusing to students, and I'm just kind of jumping around at things that I know are confusing, are the heart sounds, lub and dub. That's spelled dub. It should be dub, right? Lub, dub. Sounds better. But lub, dub. Anyway, what is the lub sound? The first noise is the AV valves. Are they opening and closing? They're just closing, right? Doors don't make sounds unless they're creaky when they open. So, I'm going to drive home this point. So, AV valve, here, I'm in the atrium right now. I'm inside the atrium. I'm going to pour blood into the ventricles. Good, it didn't creak, right? There's that bicuspid or tricuspid valve opening up. Can you hold that one? Thanks. So the valve opens up. Now the ventricle is going to contract. And the blood's going to try to push its way back into the atrium. But there's this flappy valve right there. And so when that ventricle contracts, that valve goes and closes. And that's that first noise. Now the trickier one to envision is that blood got closed down. It was trying to get out and it couldn't go back into the atria. So instead it goes out through these blood vessels, which is where we hope it goes. Then that ventricle gets done contracting and it relaxes. What could make a dub noise or the second heart sound? The semilunar valves. But why would they close? Because these blood vessels have now been filled with blood, right? I pumped a bunch of blood into the aorta, which is an elastic artery. So that aorta went, then this guy relaxes and the elastic artery tries to squeeze. You want the blood to go squeeze out this way. But it can possibly go this way, but you got little valves there and they go and they close. So the thing that causes the valves to close is lub, ventricles contract, dub, ventricles relax. And it's just back pressure of blood in through those two openings. I'll talk about the blood vessels in a minute. Heart. There's other parts. Okay, know the path of blood through the heart. I'm not going to go over that. It takes time. All right, so now let's look at these blood vessels. Here's a rather imposing list. Looks scary, right? Oh my gosh, there's so many. I mean, when you first realize that it says right and left, then you think, oh, that's half as many, but that's 2 per thing. Do remember to put right or left if it's a right or left vessel. Some teachers are more sticklers than others about that, but do your best, okay? Don't forget, at least try and say, okay, right side, left side. It's especially important, you're not gonna get any breaks around here. If you say ventricle, right? Or if you guess left and it's supposed to be right, that's wrong. But I want you to look at this list of dissection specimen vessels. These are gonna be there's some 13 or 12 of them, right? And you guys may have seen this, this diagram. If you haven't, I'm putting it up anyway. And this is simply the order of emergence of your arteries. Most of the veins are going to be, some of them have different names, most of them have the same name, right? If you know where the renal artery is, you can find where the renal vein is. If you know what the gastric artery is, where's it go? Stomach. Then you can pretty much guess that the gastric vein leaves the stomach. Arteries are going to be red, except for the pulmonary artery and pulmonary vein. They're switched. But every other time, arteries are red, oxygenated blood's red, deoxygenated blood is blue. So, I already have one. So here we go. Here's an awesome heart. There it is. Not to scale. And you've probably seen this, but I'm going to draw the aorta, and I'm going to draw some blood vessels popping off of it there. And I'm going to put the diaphragm in here. And I'm going to continue with the aorta like this. I'm going to have a couple of branches of it. I'm going to have another branch here, then I'm going to split. I've drawn this a couple times by now. But it's a really simple looking little diagram that tells you a lot. This little diagram right here is 10 out of those 13 or so arteries that are listed on there. And all you have to do is remember the order. That's it. So this big tube if I was going to draw where it was coming from, it would be coming over from here, right? So this is the left ventricle. This container squeezes blood out through this tube. What is it? This is the aorta or aortic arch. And I'm just going to abbreviate. I'm not going to write it all in there. You can figure it out later. The first branch off of that aortic arch is the brachiocephalic trunk. So a trunk simply means that it's going to split. So when you see the celiac trunk or the brachiocephalic trunk, there was another one, but I forget it. That means it's going to branch. So, and what does brachioencephalic mean? What do brachioencephalic mean?
Arm.
Brachium's arm, cephalic is head, right? So it's the trunk that's going to split to go to the arm. This is the right subclavian artery. And this goes up to the head. So this is the right common carotid artery. Then we've got a matching one over here. This is the left common carotid artery. This is the left subclavian artery. Now this aorta continues downward. You could call this whole thing the descending aorta if you wanted. For me, that's cool. If it's in the thorax, above the diaphragm, it's the thoracic aorta. If it's below the diaphragm, this is abdominal aorta. But so is this and this and this. Right? That whole tube's abdominal aorta. The first branch off, and there's some oddballs out there that you'll see them switch. You'll see the celiac come off low and go up high, and then the other one come off high and go down low. But we're not going to give you any of those on the test. So this is the celiac trunk. And now for the rat, which is where you'll get this if you get it, that's as far as you have to go, right? You don't have to know the gastric and hepatic and splenic arteries, but on a model, you do. But know that it branches off of there. That's for that left side of the page. This is the anterior. Now you may see it called the superior or cranial even mesenteric artery. But anterior on a rat, if the rat's looking forward like this is anterior forward. On a human, it's superior. Both of them's cranial. So if you really want to be safe, just say cranial mesenteric artery. But we'll go anterior mesenteric artery. These guys go to the kidneys. That's not a good kidney. You got renal disease, buddy. See, that's better. So this is the right and left renal artery. When you get down toward the hip region, they're going to split. That aorta is going to bifurcate. And these are some big arteries here. These are your right common iliac artery and your left common iliac artery. And if I was going to follow those guys over here, they would ultimately have branches that led inward. These are the internal and external iliacs. And then this one is going to go down here and eventually become your femoral arteries. So that is just the artery section. That's aortic arch, brachiocephalic trunk, common carotids, subclavian, thoracic aorta, abdominal aorta, celiac trunk, anterior mesenteric, renals, common iliac, femoral. So that's, you're only missing two, and that's the pulmonary artery, pulmonary trunk and pulmonary artery, which is a big tube that comes out like this. So that's all of them. Pulmonary trunk branches into two pulmonary arteries. So that's, I mean, it's not just no, kind of memorize the ones that are in the thorax, memorize the ones that are in the abdomen, look for the diaphragm as a dividing line, and then just count. Cool? And if it's red, I'll make sure that the blood vessels are red. If they're, oxygenated, it's not going to be some uninjected tube or anything like that. Now for the veins, pulmonary vein coming back from the lungs, common iliac, that's easy, right? Big blue thing in the same spot. This is red. There will be a blue one nearby. The big tubes that come back up to the heart, right? On this side, like this. This is the superior vena cava, or anterior and inferior vena cava. No, hopefully I'm not teaching anything new here, but maybe a different way of looking at it. I don't know. Doing what I can. Now that by the way, that superior vena cava is going to branch into some big, or it will combine from two big blood vessels that are the jugulars, right? And then that one also branches into subclavian veins, which I think you have to know. Yeah, subclavian. And that's most of them right there. Femoral vein, big and easy. All right, now when you get to the models, it's a little trickier, but try to remember where it is. Now some of these things you have to just memorize it, right? So like that circle of Willis, that blood vessel anastomosis in below the brain, right? There's I think 3 blood vessels that feed that. Your basilar, that you have to know, basilar arteries, internal carotid, and vertebral. Now, carotids, basilar, and vertebral, yeah, that's it. And they contribute to the circle of Willis. No problem. It seems like a big pile of stuff. And it's a number of different things, but it's not impossible to learn. You can do it. Have any questions about blood vessels? At the end of this, I'm going to try and roll through so that people that have to go can go. But at the end, if you want me to point out a couple things on some of the models, I'll be glad to. Hopefully I don't have to do that for 50 people, because I will do it. And here, all right, so let's talk a little bit about blood pressure. If you want to take a picture of them, I'm going to go for it. I'll draw a pie right when she's down below the horizon line. I'll go over here. So this thing looks like that Mike Wazowski from Monsters Inc., right? Wazowski, that is? All right, so cardiovascular physiology. There's some confusion often about this simple little graph. So what this is a graph, it's an EKG, right? Or ECG, whichever you want to say. of the electrical activity of the heart. Do you remember A&P1 when you talked about the sequence of events that happens to cause a muscle contraction, where you had to depolarize the sarcolemma, and then the sarcolemma would repolarize, and then you'd get the calcium, the sarcoplasmic reticulum would open up calcium channels, and calcium would go here, and then yada yada would happen, and pretty soon you'd activate troponin, and it would move tropomyosin, and then you could bond, and then you could do this, and then you could contract the muscle, right? So all of that stuff leading up to that is basically electrical activity. Tropomyosin not really, but all those ions moving back and forth. So this right here, this P wave and the QRS segment or complex and the T wave represent electrical signals that you can detect because the heart is conducting electrical activities. This is not contraction. This is not contraction. None of these are contraction or relaxation. They're all these signals that cause the contraction and or relaxation. So the P wave is what? Atrial, dipole. The QRS is ventricular, dipole, and atrial, repole. And this T wave is ventricular, repole. So basically, you're going to do that, and then you're going to go into another one over here. So these are the electrical events, and that's the terminology we use. We use electrical event and mechanical event. These are the electrical events that cause mechanical events afterwards. So this atrial depolarization causes, sometime later, sometime in here, not represented by anything on this graph, but it's going to cause the heart to contract the atria. So the P wave, that signal, that atrial depolarization, causes atrial contraction. Got me? So it's a cause and effect. The electrical thing causes the mechanical thing. This big signal is because you've got big muscles in your ventricles. This is ventricular depolarization. It's when all that sodium rushes into the heart muscle. It's going to cause, afterwards, somewhere in here, it's going to cause ventricular contraction. This is where you get those intervals coming in. So these periods are complete ventricular depolarization and period during ventricular contraction. But they're not, the contractions are not represented here at all. Ventricular depolarization, causes ventricular relaxation sometime afterwards here. So they're gonna follow. Electrical stimulus causes mechanical movement. Any questions on that? Yesterday's review was like, what? And I'm like, all right. You're okay? You can ask me if you got a question afterwards. You can ask me. I'm going to leave that up, and I'm going to erase these. All right. What's the difference between the heart sounds and the sounds of Korotkov? Korotkov sounds are the actual flow of blood that you're hearing when you're measuring blood pressure, right? So just for your edification, when you're measuring blood pressure, when you pump up that cuff, it squeezes harder than your systolic pressure at some point, and no blood gets through. You don't hear anything. So if you're listening downstream from that, you don't hear anything. Just discomfort in the person that's got 170 millimeters mercury around their wrist, around their arm. So then that cuff's going to start loosening, and pretty soon my systolic pressure is enough to squirt through. And so I start hearing that noise, and that's the first sound of Korotkov. That's basically where you measure systolic pressure. Now, as that cuff decreases in pressure, pretty soon it's weak enough that the diastolic pressure can push through also. So now systolic pressure is pushing through, and the diastolic pressure are both keeping that artery open. And at that point, you don't hear that anymore because now you just got an open tube, and it just pushes through, right? So then you stop hearing noises. So when you stop hearing noises, the lack of a sound is basically your second sound. So that's your diastolic pressure. All right, so now lymphatics and immunity, that was this week, or that is this week. I was kind of alarmed yesterday because I said, so you know that lymphatic system guy in the back? And they were like, what now? Where's that giant heavy A model? There he is. Where's the pose? Come on. All Alright, so here's this big dude. I'll come up to the front. So, this guy's super heavy. Alright, so this dude is your lymphatic system model, anyway. The white lines are lymphatic vessels, the yellow dots are little beads are lymph nodes. There's a big thoracic trunk right here, this big thing that runs right down the middle, and at the base of it, there's a swelling. It's kind of, you can't see, it's right there. There's a swollen bit called the cisterna chyli. Right, that's on there, not K-Y-L-I-E. C-H. Right, so, and all of these lymphatic vessels push this lymph upwards through skeletal muscle contraction mostly until they dump back into these little ducts right here. They dump back into the subclavian veins through these little channels. You can see them hooking over. Blood pressure up here is really low. It's like almost 0 millimeters of mercury. So that lymph basically drains back into your cardiovascular system. It's a big cleansing, cleaning fluid solution for your body. Leaks out into your tissues, gets pushed back up through these tubes, and then back into your blood supply. Now you can see some blood vessels on here. I can see the right jugular vein right there. I can see the right and left subclavian. I can see the superior vena cava. That guy exists. He'll probably be on the test. This over here, I don't know where I can go with this. Let's go here for now. That looks safe. All right, so here's Mike Wazowski. It's a lymph node, right? You'll notice that there's many little tubes coming into it. These are called lymphatic vessels. These are the same little lines that were on that guy. But you'll also notice that these vessels have little valves in them. So this valve is, the fluid can go this way, but it can't go that way, right? Can't go back. So these vessels that feed into this guy are called afferent vessels. Is this news to you, or just, if you've had lab this week, it should be good. The ones that lead away from it are called efferent. They go away. A couple of structures inside. You've got these pink ribs right here. Those are called trabeculae. The outside's called the cortex. The inside's called the medulla. These are little nodules. These are little germinal centers for various different types of white blood cell. And this is where these are your main places that you fight systemic infections. So if you get a bacterial infection, you get a lot of action in these lymph nodes. That's why mommy used to feel your neck when you were sick to see if you had swollen lymph nodes, right? Maybe they didn't know exactly what they were feeling for, but when you feel big lumps in your kid's neck, you probably are like, ooh, take him to the doctor. Excuse me, Mike, doctor's orders. Whoop, there you go. All right. Let me, I don't know what I should do. One more lymphatic system model that I think you should be aware of. Pardon? You're good? You good? Is this one? Hopefully most people have seen this, but this is a model of your, basically of the wall of your small intestine, right? So you may know that your intestine have these like villi, these little finger-like projections that stick out into the lumen. This is where the food would be to increase surface area. But these little villi have their own little capillary beds, and they also have their own little green tube, which that little green tube is an extension of your lymphatic system called a lacteal. So the villus is this thing. The green tube is called a lacteal. The whole thing represents your intestine, specifically your ileum, and this green patch right here, which you can see under microscope slides as a dark purple patch in the mucosa. This is what's called a Peyer's patch, and this is a concentrated mass of lymphoid tissue that's right at the lower end of your small intestine, right where your large intestine is, and your large intestine has a lot of bacteria, right? Most of those bacteria that are in your large intestine you don't want getting back up and populating this area, so you've got little guardhouses full of white blood cells that roam out and take care of that if that happens, right? So So you have this protection thing. By the way, these lacteals are mostly there to absorb fat because fat's kind of a big molecule. It doesn't go into your blood very well. So there's that. And that's the last chapter. Now there's a little section here called immunity at the very end. It says hapten. I'm not going to ask that. So I'm not going to put that question on the test. If your instructor went over that and said do this and that, then they might throw one on there. But if they didn't, I'm not going to put it on there. So probably don't have to worry about it. I don't think they're going to pull that one out of their hat. You guys got any questions for me? What did I miss? Thanks. All right, wait.